genome database and how we can manage that database and how we can download that sequences. Okay, so we know that there is very popular genome database, some on the NCBI, then UCSC genome browser, then Ensemble, and both three genome browser actually integrated. If some genome sequence are not available in NCBI, they will connect with you EMBL. If EMBL also lack some genome sequence, they also connect with the UCSC genome browser or NCBI. So these three are interconnected, but mostly we are using the NCBI genome browser. So today we practically learn how can we first we download the one gene sequence, then we downloaded the genome sequence. So today we actually work on the how to download any gene sequence or any protein sequence from the NCBI. Then how can we check or how can we display that gene structure? Okay, before that we need some basic idea, like what is the genome. There is some confusion in the student, what is the genome, and that genome is the level is in case of wheat it is the hexaploid. But there is the three genome, how it is originated. So genome basically haploid set of DNA content, but we know the basic sign of the genome is X. For diploid organism we call it 2X. Okay, for hexaploid it is 6X. But what is the diploid organism? You can see first Triticum monococcum and Triticum uratu, that the diploid organism, diploid wheat, and from that one genome is come, and B genome is come also the this section, and then it's make the tetraploid. So there is the A genome and B genome. But why it is tetraploid? A genome, B genome, but each genome have the duplicates. Each genome actually duplicate. So that is the tetraploid. Then this tetraploid also included the D genome. So there is the three genome, and each genome actually have the two sets. So that's why it is the hexaploid. So next we need to understand what is the eukaryotic gene and what is the function of eukaryotic gene. So this is the genome. Now we discussing about the gene structure. So you can see this is the eukaryotic gene, and what are the component of that gene? We know there is the promoter region, there is coding region, but most interesting thing is that in eukaryotic there is some intron and exon. Intron is the non-translated or non-coding region, but that intron actually included within the exon. Here you can see this is the exon 1, exon 2, exon 3. So these exon 1, 2, 3 are transcribed. Okay, but they are in the middle portion there is intron. How they are spliced out? Before that you can see this 5' UTR. Okay, this is called the 5' UTR. When before the starting the gene, that portion is called 5' UTR, untranslated region. And another thing at the end there you will found 3' UTR. Okay, now after the in the upstream, after the 5' UTR you can found promoter region, and after the promoter there is the upstream enhancer or other sequences. Okay, and so this gene this part actually operator part, then the coding region, and in coding region there is some portion is intron, and this is the stop region where, but when they transcribe, they produce the mRNA also from premature mRNA from exon and also intron. But some chemical also released by 5' UTR and 3' UTR. That 5' UTR helps in 5' capping, and 3' UTR also help in 3' polyethyl tailing. Okay. After that, after capping and tailing, that intron part actually splice out. And then finally we found that mature mRNA. And that mRNA sequence, you can see, if you sequences this part and if you sequences the mRNA, then these are not similar, because between the exon there is some intron, but in mRNA there is no intron. So we cannot go through the mRNA to your gene. So genome sequence from, but in genome sequence or gene sequence, you can found the proper mRNA. So today we will learn how to we separate that exon and intron practically from the sequences, and how we can design same type of structure, okay, using the any sequences. Okay, so let's do. First you need go to the NCBI. Just open Google page. I think you all have the laptop. So just go to the NCBI and search, write only the NCBI, not on other things, and go to the that link. There is lots of link, nucleotide blast, blanket blast, but not. We need that National Center of Biotechnology Information homepage. Okay, so we go that homepage. Interference is, you can see. Now click here, all database, and go to the gene. If you want to the gene sequence, then you can select the gene. Okay, so everyone have the laptop. Hello.
Yes, sir.
I think Sanov is talking. Other participants, have you laptops?
Yes, sir. Yes, sir.
Yes, sir.
Okay, then you just do whatever I am doing. Okay, so write a gene name like waxy gene in wheat. Then search. You can see lots of sequences, whatever is available in the NCBI, you can see here. So we can just click on that first one. Here you can see the total assemble chromosome where that gene sequences are present. You can found everything, but we need the sequences of that gene. So you just scroll down, and here you can found there is the sequences of mRNA, and another there is the sequences of protein. Okay, so now we can go through the mRNA sequence to gene sequence, or protein sequence to gene sequences. So first we download the mRNA sequences of that gene. So click on that accession number. You can found the gene bank page. Here there is the FASTA. Click on that FASTA. You can get FASTA sequence. Also you can download from here. Just click on the FASTA and click on what type of sequence you require. You just click on and it will be automatically download. But we just copy it from that sequences and keep it in one Word or text space. I prefer Word. So this is the triticum waxy gene. Waxy gene mRNA sequence. But we want to see the structure of that gene, and we physically do how to prepare that gene. You just watch. So this is the waxy gene sequence. Now you can, and another thing, we can also download the protein sequence. So we go to the back, and here you can found the protein FASTA sequence. Yeah. So you can just copy it and paste it. So this is the waxy protein. So you have the protein sequence and mRNA sequence of waxy gene. Now we go, or we found one another website that is called the phytosome. So click on phytosome. Okay, please write down all the web browser name. Okay, so this is the phytosome interference, and you can see that this is the phytosome 13 version. Okay, so now we go the tools and blast option. Okay, so here we want to search in triticum aestivum. That's why we just type triticum, and we can found there is two genome database available. One is triticum aestivum version 2.2, another is 2.1. So we can choose latest one, version 2.2, and you just take the protein sequence, just copy the protein sequence, whatever you have downloaded from the NCBI, and paste here. Please, what happened? Anything? Okay, then go. It will take few minutes. Now you can see lots of similar type of protein sequence are found through the phytosome, but we choose only whichever the identity percentage more, e value is 0, and also aligned sequence also higher. So first one, always first one is the higher, because there is the aligned number of sequence is more. That's why we choose that first one. Okay, then we just right click on the G, that G portion, and open new tab. So scroll down, and at below you can see the total genomic sequence of that gen, and CDS and peptide sequence. Okay, so from here we just copy the genomic sequence. First we copy the genomic sequence. So this is the genomic sequence GS underscore waxy gene. Now we need the CDS, that is the coding region. Okay, so we copy this CDS sequence from the phytosome, write CDS waxy. Okay, so first one is the genomic sequence of the waxy. Oxygen of wheat. Second one is the CDS sequence of the oxygen of wheat. Now we open Google, another Google page. Here we just type gene displayer, gene structure displayer server, and click on that. Here you can see they want the BED format, but we click on the sequence of FASTA. Now we put the genomic sequence of the oxygen, and CDS sequence of the oxygen. Then submit. Wait for a few minutes. and you can see this is the gene structure. Okay, so now we can change the color of the intron part. Intron are black. We just choose intron part into red, CDS part into green. This is optional. And UTR part to blue. And this size, I just change it for now redraw. So you can see this is the oxygen structure. Here, how many are exon is there? You can just count. This green color are exon. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This portion are 5' UTR. This portion are 3' UTR. And in between, the red color are intron. So this way, you can download it, the sequences, gene sequences, and you can draw the gene structure for better understanding of your gene. Now you can design your primer from the different exon if you are want to doing the real-time PCR. Okay, and you are designing some primer from the CDS sequence. So what happened? One forward primer may be here and another forward primer, a reverse primer from there. And when you do the qPCR, you have found no result or no expression. So you just concluded that that gene is non-functional, but your design of primer is wrong. So that's always you need to designing the primer within the exon for gene expression study. If you want to amplification or amplify for whole gene, then you can design the primer from the first exon to another reverse primer from the last exon. Okay, so friends, there are any question is there? Any doubt?
Hello, sir.
Yes.
Sir, can we get the PPT? Because of the network issue, I couldn't cope up with your speed, and I missed too many steps.
Because it is not, it is the live streaming. I am
Yes,
just
sir.
showing you the uh, real-time application of that software. So these are not in PPT. How can I
Okay,
share
sir.
you? If the video is recorded, you ask the organizer if there is video is recorded, then they can share. Or if you have the better connection now, I can repeat
Hmm.
it this process.
Okay, sir. Can you please repeat?
Yes. From
Yes, sir.
where you have download, you have learned the how to download the genome sequence or gene sequences
Sir, from Waxy gene, I couldn't continue with the steps. Means I just entered the Waxy gene in wheat, and then afterwards I couldn't.
in NCBI.
Yes, sir. In
Then you can get some sequences or locus
yes, sir.
like that.
Yes, sir. This page.
Okay, then click on that first one, first LOC,
Okay, sir.
and scroll down that page. Here you can see that these are the these are the mRNA sequences. Okay, and
Okay,
below
sir.
here you can found the protein sequences. These are the XP. These are the protein sequences. Okay, so
Okay, sir.
you can download any one of that, either mRNA sequences or either protein sequences. So better you can download the protein sequences. Okay, so click on that protein sequences. You can found another page where you can see that FASTA. Okay, this is the protein bank page, and you can see that FASTA sequence. Have you found this page?
No, sir.
Actually, yes, sir. Actually, after that one, I am just hello, sir.
Yes.
That location you have shown that look LOC after that sequence. I couldn't found that the below portion.
You click on this sequence. The ID is 543395.
Yes, sir.
Yes, sir.
Then wait, scroll down that page. In below portion, you can found that mRNA and proteins. There is the protein ID started with the XP. XP.
Sir, in my page, after selecting that LOC 543395, if I'm scrolling down, this sequence which is showing in your page is not coming.
Not this sequence are not coming like that. This page are not like that.
No, sir. It's like first page only.
This one.
Mm, no, sir.
Then you can click on the
This
next
one, this page.
this
Okay,
page. Then you
mm,
click on LOC.
okay, sir. I clicked.
Hello,
Hello.
sir. I have one doubt.
Yes.
So, sir, that in that gene structure which we have got from phytosome, so is it possible to assign the base pair also? Like, if we want to know, like the first exon starts from what base pair, and then further the first intron, and then followed by other exons, it starts from what base pair? So, it is possible to assign that one also.
Yes, you can. It is possible, but you need to put that sequences in manually. Okay, I can show you. Okay.
Okay, sir.
Okay. So you got that page or not?
Yes, sir, I got that page.
After that, you click on that, then you can get this type of page. I think you have the internet issue.
Yes, sir. Means after clicking that one, I am still at that same page.
Okay, then you can click on the below
Okay,
LOC
sir.
ID.
Yes,
What
sir.
happened?
Same page, sir.
So I think you have the internet issue. That's why it's need the good internet connection. Okay.
Okay, sir. Okay, sir.
Okay. So after when you are getting, I think you just watch step by step. Okay. So then you when you click on that page, that LOC ID, locus ID, you can found such type of page, and scroll down that page, you can found protein ID. So protein ID always start in NCBI. Protein ID always start with the XP. Okay. So you click on that ID. You can get that FASTA sequence after clicking. So this is the FASTA sequence. If you take the mRNA only, maybe that mRNA only for the one of the exon. So that's why you cannot get the whole gene sequence. So I think always prefer the protein sequence. So after getting that protein, okay, you just click on go to the phytosome, copy that protein sequence, and in NCBI and Google you just type phytosome, then click on phytosome. You can found phytosome 13 portal, and there is the one of the bar, that is tool, and that tools you can found some option. You go to the blast option, and you are searching the gene in triticum aestivum. So you just type triticum and choose the latest version of the genome sequence of that triticum aestivum, and here you just paste that protein sequence, and then click on the go button and wait sometimes. So you can see such type of interference. First one is the good alignment, and first one is the most of the best actually sequence alignment. So that's why we choose the first one. Okay. If you choose more than that, then you can choose through the E value. If E value is 0, that means it's good. If it is near to 0, that's also good. So we just click on the G, right click on the G. So this G means gene report. Okay. And you can found such type of interference. Scroll down. you can see there is the genomic sequences. Okay, so click here. This color here, you can see this color actually UTR, sorry, CDS region. So you just count this region. Okay. Another student asked me how we can put the base pair or length of that gene into the gene structure. So that's why we need to put the gene structure manually. So first we repeatedly do the gene structure. So that's why we just downloaded or copied that gene sequences from here to here, genomic sequences, and also copied the CDS sequences. Okay, and paste in Word file like this way. Then we can put a go gene structure displayer. You just type gene structure displayer server. You can found gene structure displayer server 2.0. Click on, and then you choose the sequence faster. Okay, and here you can paste that genomic sequence of oxygen. And CDS sequence of the oxygen. Remember these two name, actually the name of the CDS and name of the genomic sequence always be similar. If there is any error, then we cannot found any gene structure. Okay, then you just submit. After few seconds, you can get gene structure. Okay, so this is the simple gene structure, and now you can redraw. Like you can change the color or line structure of the intron, color of the CDS sequences, color of the CDS, like that. Okay, you just choose from here different color and redraw it. Okay, so then you can get this type of gene structure. Now you can save it, save the image in desktop. Okay, so another student asked me how we can put the base pair length of that gene sequences. So for that we need go to the gene report portion. Here you just copied that genomic sequence into the Word, like same as found in the portal. Okay, now you know that the total sequence length is 2898. So paste that gene structure. Now you can draw a line like this way. So this is the total length of gene. From here, you can see it is 2989 BP. So this is the total length. Now from here to here, this is the first CDS. Okay, first exon. So the length is 362, but this is the UTR region. So the length is 41. So 362 minus 41 equal to 321. So first CDS is 321 BP. This way you can put your length of the CDS also in front. Okay.
Okay, sir. Thank you, sir.
Okay. Any questions?
Sir, in that genomic sequence, some color is light blue and dark blue. No, what does it indicate? Dark blue was exon.
Oh, that's you can see here. This is the green to light green, actually UTR region. Then blue to light blue, actually CDS region, and purple color, actually UTR region. Okay.
Okay. Thank you, sir.
So this is you can this way you can design your gene structure. Now, whatever I have already discussed, if you want to design primer for gene expression study for real-time PCR, not the normal PCR. If you want to amplify that gene, total gene, then you can design the primer from the genomic sequence from here to here. Okay. But if you want to check the expression of that gene, so you can target, we know that if you want to check the expression of cDNA or mRNA, then the length of the amplification product must be within the 150 base pair. So within 150 base pair, if you choose some sequence from here, from the CDS, like you can choose the sequence from here to here. So then between that, there is lots of intron. Okay. So in that case, in real-time PCR, when you just checking the expression, you know that mRNA actually converted into the cDNA. So that's why there is a no intron portion. You cannot found any result. So you thought that the gene is not expressed, but actually expressed, you are taking the wrong portion as a primer. So always you take the primer for expression study within the CDS from here to here or here to here, like that, or from here to there. Okay. Now this is actually the gene sequences. Now if you want to promoter region, so you just watch this genomic sequence here. If that's you can see that name of that gene, the name is triticum astium whatever 4AL. Okay. This is the chromosome number. That chromosome number is 4A, and L is the long arm of the chromosome. Okay, that's why it is denoted the L. Now the sequence is in forward region. So the promoter region always the upstream of that for 5' upstream of the forward region within the 1500 BP generally. So here you can type 1500 and then submit. So you can get more 1500 sequence in upstream. So this portion actually promoter region. After the first CDS, you can take all the sequence as a promoter. Okay, copy it and paste in your Word as a promoter sequence. Okay, of oxygen. So in the next class, we can discuss how we can found whatever the promoter are available. There is the data box, what is the structure of that promoter, how many promoter is there. So in the next class we can do some practical on it. Okay, so today I will give you some homework. You can download it, or you will download it, some genes, Rubisco gene in rice, LRR gene in potato, DFR gene in wheat, MYB gene in rice. So do yourself this gene, and gene also make the gene structure of these four gene and send me. This mail ID. Okay. All are got it? Hello.
Process selecting the promoter sequence.
Your voice is noisy. Hello.
Yes.
Someone asking some questions. I don't know.
Well, now?
Hello? Am I audible?
Yes, sir. Sir, is it audible now?
Your voice is noisy. I don't know.
Yes, sir. So I wrote a message. Can you please answer that?
Yeah, sir, can you please repeat the process for promoter sequence selection? Okay. Okay. Got it. So when you are found the gene sequence, okay, then you go to the gene report, right click on it. G is the gene report. Okay, right click on it and open a new tab. So you can found the gene report. Scroll down. Here you can found genomic sequences. Okay, so click on the plus sign. You can found you can see whole genomic sequence. Now this gene actually in forward. Okay, if the forward strand, so if the gene sequence is the reverse strand, then we choose the promoter from the downstream, but here the gene is the forward strand, so that's why we choose the promoter from the upstream. Generally most of the promoter are present within the 1500 bp upstream, 5 prime upstream. Okay, so that's why here we just type 1500. Okay, then submit. So you can get one extra 1500 base pair sequences. So you just select from the before the CDS to 1500 upstream sequences and paste in the Word like this. Okay, so this is the promoter region. If a gene are present in the downstream region, then you just write that 1500 in the downstream. If the gene present in the reverse strand, then you can submit and you can found that extra 1500 sequence in the 3 prime portion. Okay, so that's so it's this way you can download it the promoter region. So tomorrow we will learn how to found the promoter and whatever the function of that promoter, what is that, and we can design that promoter structure also. Okay, anyone have any doubt? So you are requested to download these 4 gene and make the gene structure and send me individually with your name. Okay, on this mail ID. So tomorrow I will take class on the promoter and other practical session. Okay. So thank you, all of you.
Thank you, sir, for enlightening us with the valuable insights. And thank you, all the
And
participants.
students have any question, then they can share in the group also. Okay.
Sure, sir, sure. Okay.
Okay. Thank you.
Okay, thank you, sir. Thank you all. Good night.